Hi, welcome to my vlog. Can you guess what are we going to do today? Yes, you guessed it right. So here I am going to the CMU library where this video will be a tour plus an educational video. The ambience at this place is very nice where it's my favorite place to read and study. And sometimes when I get bored, I sleep here. But today, I'm going to have my virtual laboratory in the library. Hope that you will enjoy and learn on my video today. As we enter the library, we have to take note that we have to be silent since other students are studying. So before we can proceed, we have to log in first. So that's it. Finally, I am here at my favorite place to do my work. So I'm going to sit down, put my things. Oh, hi, hello. So I open my laptop, connect to Wi-Fi since our library has a Wi-Fi connection. So it's stable, so it's okay. Then go to browser. Then here is it. Without further ado, let's begin. Maybe you are wondering what kind of virtual laboratory that I'm doing. So this is the gel electrophoresis, where it is the way to sort and measure the DNA strands in tube even though you can't see or touch them. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. Performing the virtual laboratory was not that easy since first, you should have stable internet connection, and second, you should follow the procedures so that you could be successful on the entire process of the virtual laboratory. So right now, I am going to show you the step-by-step process of the gel electrophoresis laboratory. So what you see right now is the DNA strands were as big as your shoelaces. To continue, we just have to press forward, and if you miss some points, you can also press the back button. The yellow icon down there was the description of what you are going to do and the overall process of the laboratory. So this is the gel, the filter that sorts the DNA strands. We place the DNA samples into holes at one end of the gel. Electrophoresis is how we push the DNA strands through the gel filter by adding an electrical current, that is the violet one with a plus sign, and we can make the DNA moves. At this point, short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move farther away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, the DNA strands in the sample sort themselves. We also have to take note that staining the sorted groups of DNA makes them visible to the naked eye. Although we can see a single DNA strand, we can see large groups of stained DNA strands. These groups shows up as bands in the gel. And at this point, I am excited since it's my turn to run a gel where it is now my experience to do so. So, to start, we have 5 basic steps to do the gel electrophoresis. Step 1 is we have to make the gel. So, here is what we needed: an agarose, the flask, the gel mold, the gel comb, the buffer, and we also have to use the microwave. Then we have here the powdered agarose, where we have to put a small amount of agarose into our That's it. Okay. Then we have here some liquid buffer. Then we have to put some to the flask. Where our buffer is our salt solution, water solution, that will let electrical charges flow through the gel. I get difficulty some here because my cursor is not working. And yes, that's it. And now we have to place plastic wrap over the top of the flask and to prevent the liquid from boiling over. Then open the microwave and put the flask inside. Then we heat the mixture until the agarose melts into the buffer. Then we let it out. Then at this point, we have to remove the plastic wrap from the top of the flask and pour the melted agarose mixture into the mold. Then we have to place the comb into the gel on one end. The notches in the gel mold hold it in place. Then we have to let the gel cool and solidify. This usually takes about half an hour according to the virtual laboratory. So we carefully now remove the comb and leaving empty wells for the DNA sample. At this point, we have to set up the electrophoresis box. And we need the gel we have just made earlier and an electrophoresis box and another bottle of buffer. Then we have to pour the buffer into the electrophoresis box. Then we have to place the gel mold into the electrophoresis box. The gel should be just barely submerged into the buffer. At this point, we are now in step 3 and we are ready to load the DNA sample into the gel. So here is what we needed: a micropipette, a loading buffer, the DNA sample, and a DNA size standard, the gel box with buffer and gel, and also the pipette tips. With a clean pipette tip, we have to use the micropipettor to suck up some loading buffer, then add it to the DNA sample. Then we have to use the micropipettor again to transfer the DNA sample from the tube into the well of the gel. Then at this point, we have to eject the DNA sample from the pipette into the first well of the gel. Now, using a clean pipette tip, we use the micropipettor to suck up some DNA size standard. Then, we transfer the DNA size standard into the next empty well. Then, it's time to turn on the electricity and run our gel. When turning on, the black end will generate a negative charge. The red end will generate a positive charge. We plug the black cord from the electrophoresis box into the matching outlet of the power supply. Then, that's it. So, we have now to turn on the power supply. Your gel is off and running, and let's look into the gel box to see what's happening. So, we have there the bubbles that coming out of the electrodes. Repelled by the negative charge, the DNA moves through the gel toward the positive charge of the other end. Short DNA strands move through the holes in the gel more quickly than the longer strands. So, we have finished running our gel. So, for the step 5, we have to stain the gel and analyze the results. First, we have to stain the DNA in the gel using DNA staining solution, which is our ethidium bromide. So, dragging the gel out of the mold and put it into the DNA staining solution. So it takes about half an hour to stain the DNA in the gel. So because of this virtual lab, it is faster for us to do so. So we have to remove the gel, then we have to turn on the UV box, and so this is it. Now we can determine the approximate length of the DNA strands in our sample. So we have to compare the DNA sample with the DNA size standard with the base pairs. So I first did the farthest strands that goes from in our sample. So this is approximately 1,500 base pairs, and the second one is approximately 3,500 base pairs, and the nearest one is approximately 6,000 base pairs, which is my estimations are correct. So we have to take note that the estimated length in base pairs of each band in your DNA sample is based on how they lined up with the bands of known length in the DNA size standard. So we have completely run our DNA gel electrophoresis experiment. The virtual laboratory is now finally over, and I'm successfully done with it, and I'm going to keep my things and let's go out. Thank you for watching, and I hope you learned something from my video. I'm sorry if I haven't toured you around the library, since what I have said earlier, library is the place of studying, and hopefully again next time. So thank you, bye bye.
We're far away from the city lights, so small.